Okay, so it's 9 o'clock. Let's get started. So welcome everyone to our today's Zoom seminar, and especially to our guest speaker, Dr. Changtao, to spend his weekend time to share his science with us. Really appreciate it. So very brief introduction about Changtao. So Changtao currently is a full professor at Beijing University. Basic science, medical science, School of Medical Science. He's serving as a chair of Department of Immunology and deputy dean of the School of Basic Medical Sciences. So Changtao graduated as MD from Beijing University School of Medicine, mainly in clinical medicine. And then after which he pursued his PhD training still in Beijing University in physiology. So from 2009, Changtao actually moved to NIH, National Cancer Institute, study with Frank Gonzalez in NCI to complete his postdoc training. And then from 2014, Changtao moved back to China, started his independent research program at School of Basic Medical Science in Beijing University. I think a lot of us probably already very familiar with Changtao's work. The main research focus of his lab is to study the role of intestinal microecology and its metabolites in the pathogenesis of metabolic diseases and the machinery of regulation of ceramide metabolic network. His work mainly aimed to illustrate gut microbiome and its impacts on the metabolic disease. And then his lab is really pioneers a new series of cross-kingdom regulation of host homeostasis by gut microbiome enzymes, which I think we're going to hear a lot of today. And then his research has really come up with a lot of groundbreaking discoveries, including to propose a new concept of microbiome host isoenzyme. And then also his lab implicate gut microbiome enzyme as a potential target for the therapeutic intervention in metabolic diseases. Because Changtao has to leave early today, I'm not going to give too much introduction today. But I think his work has really impacted a lot of scientists in the field. And really, we are very excited to have Changtao on our platform today. And then thank you so much, Changtao, to share your science with us today. And please go ahead.
Okay, thanks, Professor Wu, the invitation and the kind introduction. It's my great pleasure to give the talk in the very famous immunology online seminar. Today, I will talk about our study about gut microbial enzyme and metabolic disease. First, I would like to introduce some background. As Professor Wu mentioned, our lab goal is to understand the key players and really underlying pathways involved in interorgan communications and how this crosstalk in cooperation with gut microbiota regulate metabolic disease. I think gut microbiota is well known. Gut microbiota is the bridge and bond between the body and external environment. And gut microbiota and their metabolites play a very important role in metabolic disease. Targeting this bacterial and related pathway would be attractive strategy for treating this disease, known as bugs for drugs and drugs for bugs. However, the main challenge in the field is most of current study mainly focused on correlations rather than causations, and the exactly molecular mechanism remain largely unknown. The main approved interventions targeting gut microbiota in clinic are mainly fecal microbiota transplantation and antibiotics therapy. For fecal microbiota transplantation, the component of fecal microbiota transplantation is very complex and increase the risk of infection. And overuse antibiotics also lead to the risk of drug resistance and emergence of superbugs. So you can see that these interventions are holistic, not specific. So our lab want to know whether there are any ways that only modulate stream function, but not affect microbiota, the whole composition. We are interested in the enzyme. So host enzyme are important human disease targets. 1/3 of FDA approved new drugs target enzymes. For gut microbial enzymes are key players in gut microbiota with functional specificity and high efficiency. So we want to know whether microbial enzyme can regulate host immunity and metabolism, and regulate host metabolic disease across the different biology kingdoms. Importantly, how to target microbial enzyme. Today, I will introduce a series of studies on microbial specific enzyme involved in bile-acid conversion. We appreciate bile-acid metabolism is what bile-acids are very important. Bile-acids are synthesized from cholesterol in the liver, and gut microbiota can metabolize the bile-acids, and primary bile-acids are conjugated with either taurine or glycine in the liver, and gut microbiota, and then metabolize these conjugate bile-acids into free forms through the enzyme of bisalt hydrolase, BSH. And secondary bile-acids are produced by gut microbiota through dehydrogenation or epimerization. For example, DCA comes from CA, and LCA comes from CDCA and UDCA. So bile-acids are very important co-metabolites of the host and gut microbiota, and bile-acids act as the signaling molecules to regulate host physiology by activating the bile-acid receptor like FXR and TGR5. Our study showed that microbial enzyme on bile-acid metabolism can help produce the various traditional bile-acids. So bile-acids act as the agonist of the receptor FXR. However, we discovered for the first time T-beta-MCA is the first natural antagonist antagonist of intestinal FXR in mouse. And moreover, we also find because TBMC is only in mouse, but not in human. And furthermore, we find a human FX antagonist is GUDCA. GUDCA is endogenous human FXR antagonist and improved metabolic disease by lowering the intestinal ceramide levels. And we also find some secondary biases like GDCA regulate the functional homeostasis of immunity cells, LC3 cells, and by TGR5 promoting interleukin-22 secretion and control the polycystic ovarian syndrome, PCOS. And HCA, high cholic acid, HCA can target FXR and TGR5 and promoting GLP-1 secretion and improved metabolic disease. Our recent work also shows that DCA improved diabetic cardiomyopathy by inhibiting CD, a very important fatty acid transporter CD36 palmitylation and fatty acid uptake through the receptor TGR5. So our previous studies show that targeting microbial enzyme involved in bile acid metabolism is an important intervention target for treating metabolic disease. However, these bile acids are traditional bile acids, and we want to know whether there are some new microbial bile acids. Important, how are they made, and what kind of physiology and pathophysiology function the new microbial bile acid performed. And due to the complex component of gut metabolites, it can lower the sensitivity and responsiveness of metabolomics. So we developed a new method, is a click chemistry-based enrichment strategy to increase the sensitivity of metabolomics. And use this approach, we discovered a new microbial modification of bile acids. This is a bile acid structure. We find that the acylation of the 3-hydroxyl group, including mono acid and binary acid conjugate forms. And further, we find a binary acid acylated bile acids we call the 3-succinylcholic acid, SUCCA. SUCCA is commonly found in healthy individuals. However, SUCCA is significantly reduced in MASH patients along with the disease progression. And we also find SUCCA shows strong negative correlation with liver injury markers such as ALT and AST levels. And then we conducted a large-scale screening of our lab set up gut microbiota strange to identify the producer of 3-SUCCA. And we cultured 315 strains, covering 97 species of gut bacteria. And we found that Bacteroides uniformis, BU bacteria, is a strong SAXA producer. And then we colonized the Bacteroides uniformis in germ-free mice and SPF mice. And we confirmed that BU bacterial colonization can produce the SAXA in vivo. And we know that the study of a biosynthetic pathway for microbial biases is a major focus and a challenge in this field. For example, for a BSH identification, take the field expert 40 years or even longer to identify the enzyme and gene clusters for these biases reactions. So what's the biosynthetic pathways of the new biases 3-succinyl in Bacteroides uniformis? And we individuals overexpressed 16 acyl transferases in BU genome based on the prior knowledge. Unfortunately, we found that none of them can produce the SAXA. So we need a new method. We use the activity-based protein purification and then perform the proteomics analysis. We get 118 candidates, and then by screening one by one, it's lucky we finally discovered a new enzyme we named BASAC, SAXA synthase, and it can produce the SAXA. And to further confirm the activity of SAXA, we use the CRISPR-Cas9 system to construct Bacteroides uniformis BASAC deficiency strain, and we find the knockout strain can't produce the SAXA. And moreover, we select related BU bacteria is Bacteroides xylem solvens BX, and without SAXA, and it can gain the ability to produce the SAXA after heterologous expression in BASAC gene. And we purified the BASAC protein and performed the enzyme kinetic assays to determine the performance of BASAC. After analyzing BASAC activity on different substrates, including biases such as CA, DCA, CDCA, and others, and we also try some short chain carboxylic acid substrate such as suc acid and lactic acid and propionic and butyric acid. And we find that BASAC only catalyze the synthesis of succinyl biacid, but mono acid biases. And we want to investigate whether SAXA because we find in clinical, we find that SAXA is negatively correlated with the MASH progression. So we want to know whether SAXA can improve the MASH phenotypes in mouse model. And we find that bacteroid uniformes can improve the mesh phenotypes, including hepatic steatosis, inflammation, and fibrosis. We find that its benefits depends on Bas-Sac activity and related SAC-CA production. And we find that SAC-CA has no effect on known bile-acid receptor, such as FXR, TGR5, and PXR. We use the spatial metabolomics show that SAC-CA is mainly restricted in gut lumen. And we found that SAC-CA improved MASH phenotypes only in SPF mice, but not in germ-free mice. So this result suggested SAC-CA benefits on MASH mainly depends on gut microbial interaction. So what's the mechanism? So we want to determine the effect of SAC-CA on gut microbiota. We performed a large-scale screening of gut microbial streams, and we found SAC-CA specifically enhanced the growth of bacterial AKK. And we also find AKK, clinical trial indicates AKK, Akkermansia failure AKK improved different metabolic disease. And we find that AKK mediates SAC-CA reverse MASH by using 2-AKK depleter. And metabolomics and RNA sequencing analysis show that SAC-CA can increase the glucose amination in AKK. So NACB is a rate-limiting enzyme, mediates the bidirectional conversion between the fructose 6 phosphate and glucosamine 6 phosphate. However, the enzyme NACB has weak amination affinity, and we found that SAC-CA can elevate NACB activity and promote amination from fructose 6 phosphate to glucosamine 6 phosphate, and promoting glucosamine production and peptidoglycan synthesis to enhance the AKK growth. In summary, we applied clinical chemistry based enrichment strategy for the first time to identify the new microbial biases. And we discovered the new 3 acylated biases. Among these, SAC-CA is widespread in humans, and it's negatively correlated with MASH. And important, we discovered the SAC-CA producer is Bacteroides uniformis, and its biosynthetic pathway, Bas-Sac, is a new synthesis of the SAC-CA biases. And we also find SAC-CA treatment can improve MASH phenotypes by promoting AKK growth through increasing NACB activity. And as mentioned before, MAFOD has become main primary chronic liver disease. With the MASH is a very important key point, easily progressing to cirrhosis and carcinoma. However, there are few drugs approved for the treatment of MASH, and clinical study indicated tobacco smoking can induce the MASH. However, the mechanism remain unknown. Traditionally, it was believed that smoking mainly caused nicotine accumulation in the lungs. However, our innovative study show that nicotine accumulated in the gut, and we discovered for the first time the nicotine degrading gut microbiota, and we identified the key strain is Bacteroides xylem solvens, and along with the nicotine and the nicotine degrading enzyme is NICX, and the degradation metabolites is HPB. And we proposed NICX containing gut microbiota would be the new strategy for MASH, mainly smoking related. And we also discovered a new mechanism by which how intestinal nicotine promotes MASH by increasing intestinal ceramide levels depend on the AMPK and SMPD3 pathway. And a mechanism study show that nicotine can active AMPK alpha-1 in the intestine, and leading to the phosphorylation of the new substrate SMPD3 at 209, and this phosphorylation inhibits K103 ubiquitination, and thereby leading the increase the ceramide, increase the SMPD3 protein stability, and increase the secretion of ceramide and promoting MASH progression. So our finding highlights the role of intestinal nicotine accumulation in MASH progression, and show that endogenous gut microbiota can degrade nicotine. And our previous work shows that gut microbial specific enzyme mediating the conversion of gut metabolites, and we also find some enzyme produced by gut microbiota may have been shaped by long-term co-evolution between the gut microbiota and the humans, and they can learn the same function as a host enzyme. So here we proposed a novel concept of microbial host isozyme, MHI. So we want to know this new concept. We want to know are microbial host isozyme wide present, and can they mimic host enzyme function and regulate metabolic disease? Importantly, can they be served as an intervention target for metabolic disease? Firstly, we developed an enzyme activity-based screening platform to identify the microbial host isozymes. This platform includes three parts. One is a stable gut bacterial culture system, and the second is high throughput screening methods based on enzyme activity, and the third one is isozyme characterization. Through this approach, we screened hundreds of human disease target enzyme, and we find 70% have corresponding isozymes in the gut microbiota. Among these, DPP4 isozymes produced by gut microbiota shows most significant activity, and host DPP4 can degrade active GLP-1, and DPP4 GLP-1 pathway is very famous, important for targeting metabolic disease. As we all know that sitagliptin, host DPP4 inhibitor, is widely used clinical diabetic treatment. However, sitagliptin's therapeutic effects show significant differences in clinical responsiveness. So we want to know whether microbial DPP4 could contribute to these variations. And we found that microbial DPP4 can be secreted, enter the host and under the leaky gut, and thereby increasing the activity of intestinal tissue DPP4, and then degrading the active GLP-1, and to promote type 2 diabetes. And through isolation and identification of human gut microbiota strains, combined with the detection of DPP4 activity, we identified 5 strains of bacteroids can produce have the strong DPP4 activity, including Bacteroides cysita macron, Bacteroides fragilis, Bacteroides agaricus, Bacteroides vulgaris, and Bacteroides dorés. And through the enzyme activity detection and kinetic characterization, we find the functional protein with DPP4 in these 5 representative Bacteroid strains. And further study, we confirmed that Bacteroid DPP4 can enter the intestinal tissue, degrade the active GLP-1 levels, and induce the glucose intolerance in mouse model. And we find that microbial DPP4 can't be effectively inhibited by host DPP4 inhibitor sitagliptin. And further clinical intervention study with sitagliptin, and we found that BT-DPP4 mediates the clinical response differences to sitagliptin. And our study showed that microbial DPP4 is a very important strategy for treating diabetes. To identify the inhibitors of microbial DPP4, we developed a high throughput drug screening system, and we find DUD4. DUD4 after screening and modification from our library of 107,000 compounds. So DUD4 is a derivative of daurinsulin, active component of traditional Chinese medicine. And we found that DUD4 can officially inhibit microbial DPP4 and increase the active GLP-1 levels and improve the glucose levels. And in summary, we proposed a new concept of microbial host isozymes, MHI. And we first developed enzyme activity-based screening platform to identify the MHI. We found that microbial host isozymes are widely present in the gut microbiota and mimic host function. Among these, microbial DPP4 isozymes as the most prominent and important. We find microbial DPP4 can be secreted into the host under leaky gut and degrade the active GLP-1, thereby induce the glucose intolerance. However, microbial DPP4 can't be effectively inhibited by the host DPP4 inhibitor sitagliptin. And this lead to the individual differences of clinical responsiveness to sitagliptin. And importantly, finally, we discovered the first microbial DPP4 inhibitor is DUD4. It's derivative of Chinese traditional Chinese medicine, and now we related sitagliptin combined with this inhibitor, and it can inhibit the host DPP4 and microbial DPP4 activity. And now the related clinical trial is undergoing. Okay, so bring us to the end. I want to thank my team members, Wang Kai, for his work on microbial host isozymes project, and Nie Qixing for his work on the novel gut microbial biases, SAXA, and Chen Bo for his work on nicotine degrading gut microbiota projects. A special thanks also to my collaborators, particularly the clinical doctors such as Professor Chao Jiang and Wang Guang and Zheng Minghua. I would like to thank you for your time, and I'm be happy to open up the discussion. Okay.